Hi everyone, these are the student instructions for the lab for PBS 3.1.5, the isolation lab. You'll start with all of the supplies, and my students and I cleaned off the tops of the tables with a bleach water solution before starting this experiment. You'll also want to make sure that you have the proper PPE, and if you have long hair, it should be tied back as well. So we're going to get started just by labeling our plates. So I have 4 TSA plates that came with the kit, and each student group will have these 4 plates. If you have 4 students at your table, each person should take responsibility for streaking one of the plates. So the first sample is going to be for patient 1. And so on the plate, I'm going to label it patient 1. I'm going to put today's date, which is 3/9/23. And I'm going to put my name as well. In addition to that information, I'm going to mark my quadrants so I know where I'm streaking. The other thing I want to point out is it's important that you're labeling on the bottom of the plate, so I have the plates flipped upside down. The reason for that is that if the top came detached from the plate and I had labeled on the top, I wouldn't know what was actually growing on the plate. So it's really important that you do your labeling on the bottom and try your best to make sure you're writing small. So that's my first sample. My second sample is going to be for patient 2, so I'll label it patient 2. Today's date, which is 3/9/23, and my name. And again, I'm going to add my quadrant markers for my streaking. My third sample is for patient 3. I'm going to put today's date, which is 3/9/23, and my name. And add my quadrant markers. And my fourth sample, in this case, we're going to use S. Marsens. So this is for the known sample. Each group will have a different known sample that they'll be working with. So this one's going to be S. Marsens. And so I'm going to put that on the plate here. And then today's date, which is 3/9/23. And my name. So each of your groups is going to be isolating bacteria from 4 samples. And again, if you have 4 people at your table, each person should be responsible for one of the samples. The other people at the table should be keeping an eye on the fire and the flame and helping whoever is streaking the bacteria with the instructions that they should be following as they go. So we'll start by lighting the flame. Just be extremely careful. You'll take the cap off, leave it nearby, and you'll light the flame. Okay, and then in my class, we're using reusable metal inoculating loops. Some of you who are watching this might be using plastic inoculating loops, and if that's the case, you'll need a new inoculating loop for each of the quadrants for each of your samples, and you won't need the flame in that case. So I'm going to start by sterilizing my inoculating loop and waiting till it turns orange. The flame, and then I'll take it out and let it cool. While it's cooling, I'm going to go ahead and get started with my first patient sample. So I have patient number 1 that we'll begin with. Okay, the process starts with me taking the lid off, and you'll notice after I take the lid off, I want to make sure that I don't put the lid down on the table so as not to contaminate the table. I'm just going to hold it in my hand. And then I'm going to begin the process by flaming the mouth of this test tube through the flame. I'm going to pass it through the flame 3 times. I'm going to be careful so not to burn the paper label, so I need to be careful and aware of that. So I'm going to run it through the flame 1, 2, 3 times, and that should kill any of the bacteria that's at the top in the opening of the mouth. I have my sterile loop. I'm going to place the loop, now that it's cool, inside my sample, and I'm going to stir it up. That way I can resuspend any bacteria that was at the bottom of the tube. Okay, and then when I think I'm ready, I'm going to pull it out, and I should have a loop full of bacteria. It's sort of like a bubble wand, and you can see the bubbles when you're using a bubble wand. Okay, I'm going to make sure that I flame the opening of the tube 3 times again. 1, 2, 3. And then I'll place the cap back on the tube. And I'm not going to need this sample anymore, so I'm going to return it to my rack. Then I'm going to take my plate that I labeled patient 1. I'll flip it over now so that the top is here. And I'm going to begin in the first quadrant by taking the loop full of bacteria. I'm just going to grab the plate open, don't take the top off completely, and I'm going to gently glide my inoculation loop back and forth, left and right, in quadrant 1. And then I'm going to close the plate and flame the loop to sterilize it. Okay, I'm going to let it cool for 15 to 20 seconds. And then after I've given it sufficient time to cool, I'm gonna twist my plate slightly, and then I'm gonna open it, and my goal is to take some from quadrant 1 and pull it into quadrant 2. So I'm gonna go back to where I was in the end of quadrant 1, and I'm gonna pull it into quadrant 2, and then I'm gonna streak into that second quadrant. Close the plate, flame the loop. Allow it to cool. Then I will spin the plate. I'm going to pick up some from quadrant 2 where I left off and bring it into quadrant 3. My goal is to get individual colonies. I'm going to close the plate, flame the loop. Let it cool. And then go from the 3rd quadrant into the 4th quadrant. So I'll pick up some here and bring it into the final quadrant. Close the plate, flame the loop. Patient 1 is complete. Okay, so we're going to move on to patient 2. I have a sterilized loop. I'm going to pick up my sample for patient 2 here. So I'll begin by taking the cap off for patient 2, keeping it in my hand. I'm going to flame the opening of the test tube 3 times. 1, 2, 3. Being careful not to burn the label. I'll take my sterilized loop, place it in the sample. I'm gonna stir it up to resuspend any of the bacteria that's settled to the bottom. I'll remove the loop. You can see I have a loop full of bacteria here. I'm gonna flame the tube 1, 2, 3 times. Place the cap back on the tube, and then return this to the rack. I take my patient 2 plate, flip it around, and then I will crab it open and gently streak back and forth in the first quadrant with my bacteria. Close the plate, flame the loop. Okay, we'll let the inoculating loop cool. I'm going to spin the plate, pick up a little bit from quadrant 1 and bring it into quadrant 2. Close the plate, flame the loop. Plate. Pick up a little bit from quadrant 2 and bring it into quadrant 3. Close the plate, flame the loop. Let it cool. Spin the plate. Pick up a little bit from quadrant 3 and bring it into the final quadrant for quadrant 4. Close the plate and flame the loop. Okay, now we'll move on to patient 3. So I'm going to take the cap off, hold it in my hand. I'm going to flame the opening of the tube 3 times, pass it through the flame, 1, 2, 3, being careful not to burn the label. I have a sterilized inoculating loop. I'm going to place it in the sample, stir the sample to resuspend the bacteria that's in the bottom. Pull it up till I have a loop full of bacteria. I'm going to flame the tube, 1, 2, 3. Place the cap back on the tube. Return it to the rack. And flip my patient 3 plate over. And I'll begin in the first quadrant, gently streaking the bacteria sample in the first quadrant, closing the plate and flaming my loop. Let it cool. Spin the plate, pick up a little bit from quadrant 1 and bring it into quadrant 2. Close the plate, flame the loop. Let it cool. Spin the plate, pick up some from quadrant 2 and bring it into quadrant 3. Close the plate, flame the loop. Spin the plate. Pick up a little bit from quadrant 3. Close the plate, flame the loop. Okay, for our final sample, we're going to do a known sample, S. marsens. It's the one that's red that came with the kit. And I'll start by opening the lid, flaming the top, 1, 2, 3. This time it's on a slant, and so we'll just take the inoculating loop, it's sterile, and put it in on the slant. Just touch it against the S. marsens, get some on the loop, pull it out, and we'll flame the top, 1, 2, 3. We'll return that to the rack. And then we will take our S. marsens plate and we'll start in the first quadrant. Open it up again, go into quadrant 1, gently go across the top, close the plate, flame the loop. Allow it to cool. Spin the plate, take some from quadrant 1 and bring it into quadrant 2. Close the plate, flame the loop. Allow it to cool. Spin the plate. Take some from quadrant 2 and bring it into quadrant 3. Close the plate, flame the loop. Allow it to cool. And then from quadrant 3 into quadrant 4. Close the plate, flame the loop. Okay, the final step will be to wrap your plates with tape. In a nice, neat package. You'll label them with your name and your class period, and we'll place them in the incubator. And we want to place them in the incubator upside down. The reason for this is if you place them right side up, you can see how there's condensation on the top of the plates. The condensation will rain down and inhibit the growth of the bacteria. So we want to put them in upside down in the incubator. The incubator is set at 37 degrees Celsius, and we want to leave them there for at least 24 hours. We'll check back in with you and let you know how many total hours they've been in when we observe the results later. Hi everyone, these are the instructions for the PBS 3.1.5 isolation procedure lab. If you've watched the beginning part of the video, you know that we've gone through procedure day 1 on how to streak the plates for isolation. If you were absent, you might want to run through this virtual streak plate lab to practice the process. Just remember the steps that happened on day 1. Today we're going to focus on what happens when we observe the plates on day 2. And by the way, this could be actually day 2 or day 3 or day 4, depending on how your teacher has this set up. I think the perfect sweet spot is on day 2 after doing the isolation. So after 48 hours, we saw the best results. We conducted the isolation streaks on 3 patient samples, and we're now ready to observe the results. If you are successful, you should see individual colonies growing on the agar plates. We'll be using those individual colonies to do the next step of testing in 3.1.6 when we do gram staining. You're hopeful that through observation, you'll be able to identify one organism that's common between the patients. If you're working on this in class, make sure you have your PPE on as you take the plates out of the incubator. When you remove the plates from the incubator, be careful not to open them. You'll be observing the growth on your plates. If your team created successful isolation streaks, you'll see the isolated colonies. You'll see these individual dots here. These are the colonies that we'll be using in the next step of testing. If you didn't have results, not to worry. I have some results posted here that you can use. So these were my results after 24 hours. So I'll pull these up so that you can look at them here. Okay, so I have patient 1, patient 2, patient 3, and I have the 3 known samples, B. megatherium, K. rhizophila, and S. marcescens. So you can check out those results at 24 hours. I also have a picture after 48 hours, which I believe is the best time frame. And they're set up in the same arrangement. So patient 1, patient 2, patient 3, and then the 3 known samples, B. megatherium, K. rhizophila, S. marcescens. And then I have it after 72 hours. Not much more of a difference here, but you can definitely use my images if you were out or just as a comparison. All right. The other thing that I'd like to just stop here and point out is that the results that we got were not the expected results for my students and I. So Project Lead the Way provided some pictures of expected results, and we're going to use those as we move on here as a comparison so that we can continue on the assignment. So I'm going to open these up so that you can look at them as well. Okay, so here's the patient 1 close-up, the patient 2 close-up, and the patient 3 close-up. We did have yellow colonies in our results and the white colonies. We never saw any of the red colonies, and even after a week's time, we never developed any red colonies. We never saw the color, even in the known samples. So that happens sometimes, and we will just note that and move forward on using these expected results and then our results as well. So different species of bacteria will often look different when grown on the plates. Some could be colorful. We saw some yellow ones, we saw some white ones. Some could grow large, greater than a diameter, or they could be small, less than 0.5 millimeter. They can grow in different shapes, forms, and have elevations or heights. Their edges or margins may be different. And so here are some of the words that we use to describe the forms, elevations, and margins. If you have the plates in front of you, take a few minutes, use a magnifying glass, and look at the colony diameters. We're going to be recording those results in just a few minutes. There is a reminder here, do not open the plates. So what I would like for you to do is we're going to screenshot the results for the 3 patients. Your teacher may have you also draw the results. In my class, we're going to be screenshotting the results from the expected results from Project Lead the Way at this point. So I'd like for you to screenshot the patient 1 close-up, the patient 2 close-up, and the patient 3 close-up, and insert those images into your document here for patient 1, patient 2, and patient 3. The next question asks how many different species of bacteria appear to be in each patient sample, and how do you know? We're going to use the Project Lead the Way results for this because they're the expected results to answer these questions. That'll help us move forward, but don't worry, we'll also be using the images that we took of our own plates and then making a comparison. So in patient 1's sample, if you zoom in, you can see that there's white colonies or cream colored colonies and yellow colonies as well. In patient 2, you can see that there are some yellow colonies and red colonies. And in patient 3, there's again yellow colonies. And if you look closely, there's also white or cream colored colonies in patient 3's sample. So at that point, you should be able to explain how many different species of bacteria appear to be in each patient sample. And how you know is because there's different colors of those bacteria, which indicates different species. At this point, we'll move on to this table, and in patient 1, we want the color of the colonies and then also the form, elevation, margin, or size. For size, we'll just use the words large or small. Large if it's greater than 1 millimeter in diameter, and small if it's less than 0.5 millimeter in diameter. For the words for margin, elevation, and form, I'll remind you that you have a list of descriptions here. And the colors we said were from this picture here. We have in patient 1, we have yellow and cream colored. In patient 2, we have red and yellow. And in patient 3, we have yellow and cream colored colonies. So take a few minutes to take care of that. Is there one organism that's common between the patients? We can see here that the one organism that's common between the patient is the yellow colored colonies. That also was the case with our plates and our own results. We had yellow colonies in each of the 3 patient samples, patient 1, patient 2, and patient 3. So our results do track for the one that's in common. And then it says here, try to determine the species of bacteria on each plate, match your observations with the culture key. This table lists some of the common causes of nosocomial infections. So E. coli, the color of the colonies were cream. You could see that the one that our patients had in common is a yellow colony, so we can rule that one out. The one next, Staph aureus, is yellow, circular, convex, entire, and large. And when I look at these colonies here, that seems to fit. So I'm going to go ahead and highlight S. aureus as one of the possibilities. So here. Okay. Our colonies were definitely not blue-green or brown or tan or cream, but this S. multifila does look like it matches, so yellow, circular, convex, entire, and large. So we'll go ahead and highlight this one as well as a possibility. Okay, so it says compare your observations to the results the hospital lab obtained for the other 8 patients. Remember, there was 11 total patients. And so for patient 4, we have another set of colonies. It's yellow, circular, convex, entire, large. For patient 5, we do as well. For patient 6, yellow, circular, convex, entire, large. You'll see for that each of the 11 patients, that same colony that's yellow, circular, convex, entire, large is present. which leads us to believe that this is the culprit for the infection. So based on your observations, what are the possible identities of the organisms causing the hospital outbreak, and how do you know? If we look back here, we have highlighted the 2 possibilities, and you should be able to describe how you know in that answer. All right, so gross examination of unknown bacteria is generally the first step performed in identification. While it's helpful in eliminating some possibilities, it's rarely sufficient alone. This is due to the large amount of bacterial species and degree of variation between them. So you will be storing those plates back in the incubator to be preparing for 3.1.6 when we do gram staining of the yellow colonies. You should definitely use aseptic technique to clean up your lab area. Remove your gloves, make sure you wash your hands. And before you put everything away though, I do want to have a picture of the plates that you created here. And so those of you that are in class, you can take a picture of your plates before you put them back into the incubator and insert them here. Make sure I can see the labels and the individual colonies if you were able to be successful in your isolation procedure. So make sure the pictures are clear and you put them in here. If you were absent, you could always use my example pictures that I provided in the document. So you can screenshot these and put them in your document here. You'll see that we'll have another submission on another day after we've done the gram stain results here. Now, some of my most observant students recognized that the 3 known examples of bacteria that we streaked plates for, B. megatherium, K. rhizophila, and S. marcescens, were not on this list of common nosocomial infections. And if you were one of those observant students and you're wondering what the deal was with that, I'll give you a little peek behind the curtains here. And it turns out that since these are nosocomial infections, it's not something we'd really want to have in class. And so what's happening here is that these bacteria are playing the role of other bacteria. So B. megatherium is cream colored. It's playing the role of possibly E. coli or this one here. And then there wasn't even a red example here in our list. And then the one that we're really concerned with, the one that we'll be using in class is the K. rhizophila example. And you'll see like all of our patients have that. So K. rhizophila is playing the role of either S. aureus or S. multifida. And it has similar colony characteristics, and that's why we're able to use it, and it's pathogenic, and so we were able to keep that in class and not cause people to get sick, and so it's just playing the role of S. aureus and S. multifida. Hope that helps in your determination as we move forward. If you have any questions, feel free to reach out. Thank you so much.